So welcome back. I'm Bhavna Muralidharan. I'm a faculty at INSTEM, and I'll be chairing this session, which has a very interesting lineup of speakers and talks. Our first speaker is Dr. Pedro Rocha from NIH, and he'll be talking about untangling enhancer-promoter interactions throughout mouse development. Over to you, Pedro.
Thank you. Switching laptops. Thank you so much for the invitation and for organizing. Can you guys hear me in the back? I feel like, okay. It's not the microphone, right? It's just me screaming out loud. All right. They'll adjust, yeah. All right. But everyone can hear me. That's what matters. Thank you so much for the invitation. Thank you for organizing such a beautiful conference. I look forward to this week and for having my slides. Here they are. So we're the unit on genome structure and regulation. Like Dan, we're also at the NIH. Like our name says, we're interested in understanding how the genome is folded inside the nucleus, how that contributes to regulate its expression, and how that's important to specify cell-fit decisions and to build mammals. And the reason why we study nuclear organization is because gene expression is often controlled by enhancers that are found at very distal, very far distances from their target genes. And so studying the mechanisms that guide these distal enhancers to the target genes, and equally as important, which mechanisms ensure that interactions are not miswired and that and that off-targets are not activated are equally as important. In the first years, our lab has been focused on a very specific type of mechanism that folds the genome, and that's loops formed by the action of CTCF and the cohesin complex and friends. This is known as the loop extrusion model, where CTCF is a transcription factor that binds DNA in a way that it's determined by its motif, and that also determines where its N-terminus is going to be placed. Cohesin is a ring-shaped complex that is loaded onto interface chromatin. It can extrude DNA through its ring, and it will stop extrusion once it bumps into CTCF with the N-terminus pointing towards the extrusion direction. So the voice is going up and down, right? Yeah. I'll keep screaming when I realize. And so that means that if you have convergent CTCF motifs pointing at each other, that is able to maintain a stable loop, even if, as we now know, these stable loops don't live for that long inside the cell. If you don't look at high C maps all day like we do, this is how CTCF loops can look in a high C map. That corner over there, that strong signal represents a strong frequent interaction between the two anchors of where CTCF binds. And these bases of these triangles, that's where each anchor is, and they represent regions of high insulation where interactions occur more seldomly across where CTCF binds because of the formation of these loops and formation of insulation. One of the best things about this model is that it gives you a very easy, even if very simplistic way to think about how mechanisms of nuclear organization can directly impact gene regulation. So you can easily think that if you have CTCF motifs between an enhancer and a promoter, formation of a loop can bring the two together and drive transcription. And in contrast, if that same motif is found between the enhancer and the promoter, formation of the loop is going to result in insulation and not transcriptional activation. Of course, a question that many people have asked is whether these models explain gene regulation in vivo. There's evidence for it occurring through these two models, occurring in vivo. Sometimes yes, sometimes no. So the answer, I think, from the work of many labs, including our own, and probably an answer that it's given for almost every mechanism that regulates gene expression, is a resounding "it depends." And it depends on many things. It depends on genomic context. It depends on cellular context. And that's exactly what our lab is trying to work on, in trying to define what are the different contexts that modulate how nuclear organization can regulate gene expression. In other words, we're trying to understand whether nuclear organization impacts gene regulation differently throughout development and what distinguishes these different concepts. So one of the approaches that we use to study this question is to perturb the nuclear architecture of loci that code for transcription factors that are important for different developmental processes, so that we can study their impact early in development, but also at later stages. And the person behind this specific approach is a postdoc in our lab, Shrita, who's here in the conference. She's speaking tomorrow, who's getting help from our lab manager, Nina. One of the loci that we decided to use to employ this approach is the SOX2 locus. We like it because it's in a gene desert region. It codes for a transcription factor. It's expressed in a variety of different tissues, usually by different enhancers. And in many of these cases, it's a one-to-one relationship. For example, in the epiblast, in mouse embryonic stem cells, most of its expression is controlled by this large enhancer of 10 kb located more than 100 kb away, known as the SCR. Shrita showed that if you knock out SCR, basically SOX2 expression in blastocysts is almost fully gone, and that these embryos cannot implant, showing that this basically recapitulates the loss of SOX2. When you look in mouse embryonic stem cells, you see that they interact at strong frequency. You have CTCF binding here, CTCF binding there. There's the strong interaction, formation of a domain, and that it's nicely insulated from neighboring regions. So we can use it to try to understand better what defines the ability of nuclear organization to impact gene regulation. In other words, here we're specifically asking what defines the ability of CTCF loops to block enhancer-promoter communication. And we use a very simple conceptual approach, which is we insert CTCF motifs between the enhancer and the promoter with the hopes that this would block cohesion coming from one side and separate the enhancer-promoter pair. This works really well. When we inserted the CTCF motifs between the SOX2 gene and the enhancer, we form here a new boundary. We, in fact, created a allelic series of mice with different insertions, motifs to the left, motifs to the right, the double motifs left and right, and an insertion where we had 18 motifs. This is all published, so I'm just going to summarize our findings. We saw that enhancer-promoter contacts are better blocked when we have two loops, one pointing at each side of the interaction pair than when we have, for example, 18 motifs all pointing to the same side. We also saw clearly that the frequency of enhancer contacts determine transcriptional output. So when we sorted the mouse lines by how much insulation they have, you can see that the ones that insulate the most, it's also where SOX2 expression is reduced the most. So CTCF can block the ability of enhancers to activate their target genes. But you can also see that they do not block as much as the full enhancer deletion. So even with this very strong double boundary, you still have some induction of SOX2 expression, and you can see that they still interact even if at low frequency. So SCR can still activate SOX2 across CTCF loops, and all these mutant mice were still able to implant. This goes with what Dan was saying this morning. Mouse embryos go to the work of making this much SOX2, but even if you lose 90% of it, they're still fine, as long as they have some of this SOX2, which is an essential transcription factor. But the amount of SOX2 doesn't really matter. So, as I say, we like to see what happens to these modifications throughout mouse development. SOX2 is a great example because we can look at, for example, what does it do later in development, specifically in the neural specification. And as you can see, even in mice with the strongest insulation, where we put these double cassettes, SOX2 expression in neural tissues was not affected at all. And in this case, SOX2 expression is controlled by enhancers that are even further away, more than 500 kb. So does this mean that SOX2 can always bypass CTCF insulation and get activated by distal enhancers? Not exactly. When you look a little bit closer, you see that at this stage, at 9.5, expression of SOX2 in this little tube over here is completely gone in these mouse embryos. This is the anterior foregut. It gives rise a little bit later in development to the esophagus, the stomach, trachea, and lungs. But in these mouse embryos, rather than just reducing a little bit here, the insertion of CTCF motifs completely blocked the ability of SOX2 to be expressed. And these mice, rather than having two tubes, the esophagus and trachea have a single tube that separates into the lungs and to the stomach, and therefore they died very shortly after development. What was interesting for us was to try to understand why, unlike neural tissues or Unlike the epiblast here, insertion of these CTCF-mediated insulation completely blocked SOX2 expression. So we looked at what is potentially driving SOX2 expression in this case and compared it to the cases where the effect was not as strong. You can see that in tissues that come from the anterior foregut, when you look at enhancer marks, rather than being concentrated in a small, dense region full of enhancer activity, they are spread out through a larger region. So what we proposed was that in mouse embryonic stem cells, we have formation of loops. This is able to separate SOX2 from its distal enhancer, but this effect is not absolute, and some SCR still interact with SOX2 enough to drive the small amount of SOX2 that is required for implantation. In the anterior foregut, these enhancers are distributed to a larger region. They are not as clustered, and maybe this means that they are not as good as bypassing CTCF-mediated insulation. So in summary, what defines the ability of CTCF cohesion loops to block enhancer-promoter interactions? Perhaps not surprisingly, the two sides of this equation play an important role. The stronger the boundary, the stronger the effect, and we show that divergent CTCF clusters are better insulators if you have CTCF motifs pointing in both directions to separate enhancer from promoters. And obviously, the robustness of the ability of the enhancers to find their target genes. And we propose that regions that have a higher density of enhancer elements may bypass CTCF insulation better. So we spoke here about the enhancer recruitment, enhancer insulation role of CTCF. So let's mention briefly the role of CTCF in recruiting enhancers. SOX2 could be the perfect example of how CTCF is used for this, because as you can see, CTCF binds just upstream of SOX2. CTCF anchors tissue-specific interactions with enhancers that drive expression of SOX2 in these different cells. So for example, in neural tissues, you have CTCF binding here, you have this distal neural element, you have a nice interaction over there. In the epiblast, what we've been talking about, this interaction over here seems to be anchored by CTCF. But when we removed CTCF in all these tissues, we never saw any effect on SOX2 expression. So at least in these cases, CTCF is not required at all to recruit these distal enhancers, and the enhancers find their way to their target genes through different mechanisms. As I'll show you in a little bit, this does not mean that CTCF never plays a role in enhancer recruitment. So to define what contexts allow nuclear organization to impact gene regulation, we try to predict, for example, which domain boundaries are essential for gene regulation. And one specific question that we try, or one specific hypothesis that we tested was whether domains that have multiple developmental regulators may be more susceptible to perturbation. And we found a very nice locus to study this question. This is at the end of mouse chromosome 7. We have 3 different FGF genes, all separated by clusters of CTCF motifs. These FGF genes all have different functions and different expression patterns, which means it was a great tool, a great model for us to address this question. I'm not going to go into details at all over here because this is what Shrita will tell you about tomorrow, but suffice just to show you that, I just want to show you that if you delete a single CTCF motif within this cluster that separates the FGF genes from this upstream gene, you go from 0 to a lot of FGF expression. And in this case, we have 100% lethality with just the loss of a single CTCF motif that separates these FGF genes from an upstream enhancer. So in fact, sometimes when you have these multiple developmental regulators inside a large domain, this might be enough for disrupting gene expression quite a bit. So going back to our question, can we then predict when is CTCF essential for gene regulation? I showed you cases in SOX2 where there's a lot of robustness to perturbation. In the FGF case, that, as I just very briefly showed you, that is the opposite. How can we predict when it will be essential? We're in the age of degron and rapid degradation and quickly assessing the impact of rapidly degrading proteins and seeing the genome-wide effects. I think those approaches are really good. They also suffer from a few problems of leakiness or incomplete degradation. And so there's also value in this kind of approaches that we do where we perturb just one region of the genome and measure the impact in cis of regions next to where we perturb. So we wanted to develop a high-throughput method to study the cis-direct roles of CTCF on gene regulation. And what we do here, we developed a technique that we borrowed from the Bas van Steenzel lab, where we use a reporter, where expression of GFP is being driven by a minimal promoter whose activity is controlled by endogenous enhancers. Then we put a CTCF motif flanked by LOXP sites. We insert this into thousands of cells, and so we end up with a pool that has 10,000 different transgene insertions in different sites throughout the genome. We take this pool and we activate CRE in part of the pool, and we separate it, and the other half of the pool does not get CRE. But the two halves have the same integrations. But in one of them, CTCF is still there, the other one, CTCF is gone. Then we map integration location, and we measure reporter expression. I don't have time to go through the details, but as I said, this is published. This is from, the method is published from Bas van Steenzel's lab. He uses a barcode composed of just 16 base pairs, but that means we have way more barcodes than cells, so each barcode integration is unique. What we do is from the pool of cells, we isolate RNA and DNA, and we count barcode molecules in both RNA and DNA so that we can have a normalized barcode expression. Because we have that barcode, we can also use the DNA that we isolate from these cells to map where each transgene is integrated. Does this work? Yeah, it works pretty well. So each circle over here is one integration where we know where it integrated. We know how much the reporter is expressed when CTCF is present versus when the CTCF is gone. We also compared what happens when we use one CTCF motif in the cassette versus 3 CTCF motifs. You can see that we find regions where we have higher expression without CTCF, so you can see that CTCF is acting as an insulator in these cases, and areas where CTCF is where expression is higher when CTCF is still present rather than being deleted. So in this case, CTCF seems to be working as a recruiter of enhancer activity. You can see here how the absolute fold change is higher when we have more CTCF, showing that there's a dose-dependent effect here, which makes sense because CTCF, just like any transcription factor, has to bind, unbind. So if you have more cassette, you have more probability for it to bind and recruit cohesin and form loops. We confirm that the measurement of expression from the barcodes that we get from our approach, if we measure the expression by qPCR, the results are pretty much the same. And then we wanted to see, are these things actually affecting chromatin the way that we think they're affecting? So we isolated clones with specific integrations, and then we did capture micro-C using probes to the reporter elements that we integrated throughout the genome, because that part is unique, does not happen anywhere else in the genome. So you can see here the interactions of a reporter that integrated next to this gene that codes for an element sometimes of polycom. You can see how it does not engage in much interactions throughout the locus when CTCF is deleted, when we add 4HT to remove the CTCF motifs. But when CTCF is present, and it's integrated in the positive orientation, it can find these CTCF motifs on this side that are in the negative strand and form strong loop over here, which increases the ability to interact with these enhancers. And it even leads to a very specific interaction with the promoter of the gene that is next door to where it integrated. It doesn't lead to a very high upregulation. It's a twofold change in gene expression, but it's driven by this small amount in interaction with enhancers. This was a small case. I think I really I would like you to appreciate how the interaction is increased with the promoter over here because of the interactions increasing with the enhancers over here, but we also have some more dramatic effects. This is another gene where we integrated reporter a little bit further away, and you can see how the integrations in red without CTCF, again, nothing is going on, but if CTCF is present, it starts engaging with the CTCF motifs downstream, and here it interacts way more with the enhancers that are found just upstream of where integrated. And in this case, we have a much stronger effect on gene expression. So yes, CTCF is able sometimes, in some cases, to recruit enhancers to drive expression, and that's very clear. So we expanded these pools. We now have just finished getting data for more than 50,000 different integrations throughout the genome. We're still in the process of analyzing what drives some cases to be sensitive to the presence of CTCF and some cases not. You can still see how it seems like, interestingly, most of the times, like 50% of the time, or 97% of the times, we don't see a statistically significant effect of the presence or absence of CTCF. But in other cases, we see stronger impacts. So for example, over here, we see 0.6% that cases where CTCF insulate and 2.9% where CTCF seems to induce. We're still in the process of analyzing this, but something that we noticed was that a lot of the data seems to be on the lower end of the distribution, where we don't have so much expression of these reporters. So we decided to test if this is something that could determine whether CTCF could impact gene expression or not. And so we repeated these trip experiments, but we swapped the promoter that is driving expression of the reporter. So rather than just always using this minimal promoter that has very weak activity, we decided to try stronger promoters and see what would happen. So we tried, or promoters of different activity. We tried the SOX2 promoter, the GAPDH promoter, a housekeeping gene, and also HPGK promoter, which is a very strong promoter. These are integrations of these reporter cassettes throughout the genome. You can see, perhaps as you would expect, GAPDH is the weaker, but it's obviously depending on where it integrates. There's a big range of expression. You have the minimal promoters, the weakest, and SOX2, GAPDH, HPGK. When we look at the effects of having these promoters, you can see how the minimal promoter is the one that has the strongest variations of gene expression, or the strongest ability to react to the presence or absence of CTCF. That's also quantified over here. So weaker promoters are more sensitive to perturbation or to be regulated by CTCF. In other words, if you're an enhancer that drives strong expression of a certain promoter, that promoter will always be able to be activated by that enhancer, whether or not CTCF is there. It might be that in our case, because the CTCF is so close to the promoter, insulation is probably being bypassed more easily. But one thing's for sure, if it's strong expression, then CTCF's impact will be smaller. So in summary, for this part, CTCF has minimal impact on gene expression at most genomic loci, but sometimes this effect can be dramatic. We show some cases both as a recruiter and as an insulator. We showed that if you have more CTCF, the effects on gene regulation is going to be stronger. We also show that lowly transcribed promoters are perhaps more sensitive to the action of CTCF. Now, just to end, this is the last slide. If I lost you throughout the presentation, but if you're interested in finding target genes, that's where nuclear organization is at its best. One take-home message, or two take-home messages that I want you to take with you is that interactions within domains will always be favored. So if you find an enhancer, you're trying to understand which genes are perhaps regulated by this enhancer, always think that interactions within the domain will be favored. But some enhancers can cross boundaries. Some enhancers are better than others. Some boundaries are more permeable than others. We're still trying to understand this better, but I think we're making a lot of progress compared to where we were a few years ago. So I presented the work from Shrita. She'll tell you all about the FGF story tomorrow. I think there's a lot of interesting aspects for you to learn. Nina helped in a lot of those experiments, and this is the rest of the lab. This is our collaborators, and I also want to thank you for your attention. If you have any questions, I'll take them.
Thank you so much, Pedro, for this lovely talk. I think I'll go to the audience and then.
Thank you. Here, very nice talk. So I was curious in the last part where you look at all the statistics, if you are able to look at the genomic distance between promoter and enhancer, and if you see an effect of the genomic distance.
Yeah, so that's exactly what we were doing. We did it when we still had very few numbers of integrations. We couldn't see something very specific. We had very few. Now that we have these 50,000, that's exactly the kind of experiments that we want to do. See how far can they be. Yeah, but we haven't done that since we got these larger numbers.
So would you okay.
Yeah, go ahead.
Yeah. So would you expect, yeah.
Sorry.
So do you expect any active mechanism by which CTCF might not quite localize or organize regions which are at the telomere or the centromere?
Could you repeat the beginning? I couldn't
So
understand.
the organization of telomere or centromeric associated regions which are more condensed. So would CTCF really function in those domains of the genome?
I mean, if you have CTCF motifs, and CTCF can bind even in regions that are silent, right? I think if they are very silent, then you will need only the very best motifs will be able to bind CTCF. In terms of recruiting enhancers and so on, obviously there aren't as many enhancers there. But those regions are also organized, right? So extrusion happens throughout the chromosome. So I think most of the mechanism would still work, but it's different from what we're talking about here. Yeah. You can scroll.
Non-active. Okay, there it goes. Really clever, Pedro. So I guess I have a technical question. Do you ever worry about the penetrance of the Cre recombinase? I mean, how do you ever get false negatives where you just didn't cleave out the CTCF, and how do you find those?
We assessed it through a variety of methods. So we integrated a reporter, like a lock stop reporter, just to see how many cells actually don't make the recombination, because it's a similar cassette what we're doing. And in our system, basically 97% of the cells or something excise within 24 hours. Then we also do, every time we do this, we do qPCR on non-recombined compared to recombined, and it's similar to the FACS results of 95 to 97% of the cells recombine very quickly. So I think it is recombined.
Pedro, here. Very nice talk. So in your system, so considering that if CTCF is the only boundary element, it makes sense. What about non-CTCF elements? What we know, it can also halt the loop extrusion, right?
Yeah, you're talking about specifically...
Yeah, any other non-CTCF boundary elements.
Yeah.
So if you consider that, what happens?
Yeah, we're doing trip in a bunch of things. Everything that is a cis element,
yeah,
we're testing it. There aren't that many. There's a lot of things that we know can create boundaries.
Yeah.
There aren't that many things that we know the sequence that will recruit it. But there's a few, and we've been testing them. And there's things we have that we put in between the LOXP sites that recruit enhancers way better than CTCF.
Okay, thanks.
But we're still working on that.
Did you say that weak enhancers are more sensitive to CTCF? And so how do you quantify the enhancer strength?
Sorry, I meant weaker promoters. Weaker promoters. So integrations where we have lower expression, or if in case we swap the promoter cassettes, promoters that are not able to drive such strong expression, those are more sensitive to the action of CTCF. That's what we saw. Well, in our case, we quantify by looking at the reads. So here, each circle is one integration, right? So obviously, because we're integrating randomly throughout the genome, if it's closer to an enhancer, it's going to be more expressed than if it's further. But you can see at the distribution of all integrations from one specific cassette, how this is weaker than what this achieves, or this and that. And you can see how the PGK, which is probably the strongest promoter we have, that drives the strongest level, and it's also the smallest distribution.
Sorry, can I just add to that question and ask, what is the sequence of, is there any merit in looking at the sequences of the weak promoters and the strong promoters, and is there any motive for elements?
Yeah, I think there's a lot of interest. It's not necessarily the kind of work that we do in the lab, but I think there's a lot of work to understand what's the language of promoters, for sure.
Okay, yes, thanks. Dimple, and then after that, Pradeep.
Okay, very nice talk, Pedro. So I was wondering this idea of multiple weaker CTCF motifs together acting as a better insulator or boundary. Have you tried to, change the strength of these motifs?
No, that we have not. That could be done. That, like, we actually, let me remember which motifs this was already some time ago. I think we put here the same motifs that are in Shrita's paper. So these are very strong CTCF motifs that bind a bunch of CTCF no matter what. It would be interesting to test different ones because we do know that here we're taking the core 50 base pairs plus 60 base pairs on each side. So it takes basically the entire motif, but it would be interesting to test motifs of different affinity and see how that could affect things differently.
Sorry, I think you will have to hold. Pradeep, one last quick question.
Yeah, so that's what we're trying to do here, right? So when we look at this side of the, I think I had. Yeah, so all of these are sites where CTCF is inducing expression, where expression is higher if CTCF is still present. So we're trying to define why does that happen? Why are these sites much more sensitive and why these don't care so much about it? It could be a variety of different things. It could be what Daniel was just saying, that these guys are at a distance where it can still act, or if it's too close, it won't be able, CTCF impact won't be determined. So it could be the type of enhancers, could be distance, it could be what other transcription factors are present compared to what can bind the minimal promoter. It could be a variety of different things that we're still trying to understand. We have a rich data set now still to explore.
Okay, thank you so much, Pedro. Let's invite, round of applause, please.